is the nucleus. Okay, the nucleus, the largest organelle found at the center of the cell is called the nucleus. Okay, so has nuclear membrane, because we are talking about the eukaryotic cell, so that each organelle has their own membrane, so that membrane-bounded organelles are found in eukaryotic cells. Okay, so the nucleus is organelle, okay, or one of the organelles, and has its own membrane, and that's called nuclear membrane. Okay, but still there is communication between the nucleus and the cytoplasm because of the nuclear envelope. So nuclear envelope is a whole-like structure, so which the nucleus is communicated with the cytoplasm of the given cell. Okay, and the main function of the nucleus is to control the cell activity, or to control the cell activity or the cell functions. Okay, and that is because it contains the genetic materials, not found at the center, not because of its largeness and so on. No, that is because of the content, or because nucleus contains genes. So genes are molecules found on the DNA. Okay, the DNA. Okay, and that. There are many genes. Actually, there are many genes found in each DNA. Okay, and each gene has their own triplet code word. Actually, you discussed about protein synthesis in your biochemistry when you are going to discuss about the protein metabolism. Actually, so don't worry about the protein synthesis. Or how the DNA is going to replicate and so on. Don't worry about this one, because you'll have courses which called protein metabolism, and you'll see the concept in detail. But you should know that, at least you should know that nucleus controls the cell activity because it contains the genetic material. Okay, the genetic material, or the genes. Okay, and the function of this triplet, or that's called the 3 sequence. Okay, like for instance, you see the gene 1 has triplet codon sequenced as C and that AG. Okay, and if you see the gene 2, these are actually on the DNA. Okay, and you can have the gene 3 with a different sequence, so that the different genes, okay, have different sequence of triplet code word, so that different genes involved in the synthesis of different proteins. Okay, and those proteins can be ion channels, those proteins can be enzymes, those proteins can be receptors. Okay, so that the cell activity is going to be controlled by those proteins made by because of the DNA. I think you know the information in the DNA is transcribed into RNA, and that's called transcription. And the information transcribed from DNA to RNA is translated into the transfer RNA. Then that's called translation. So and finally, different proteins are synthesized in ribosomes. Okay. So the first nucleus, the first organelle is nucleus, and its function is controlling the cell activity. Okay. The next organelles are ribosomes and endoplasmic reticulum. So ribosomes are organelles found in given cells. Okay. So those ribosomes may be found freely inside the cytoplasm without combined with other organelles, and they are called free ribosomes. And some ribosomes combined with endoplasmic reticulum, and those are called bounded ribosomes. So ribosomes can be free ribosomes and bounded ribosomes, but both the free and the bounded ribosomes are very important to synthesize what? Proteins, because actual amino acids are sequenced in ribosomal RNA. You can have messenger RNA, you can have transfer RNA, you can have ribosomal RNA. So ribosomal RNA are found in the ribosomes where actual amino acids are sequenced. Then you can have polypeptides. Those polypeptides are proteins. But the proteins synthesized in bounded ribosomes and proteins synthesized in free ribosomes are not the same. For instance, if you see proteins synthesized in the bound ribosomes, leave the cell to act somewhere in your body, and that's called exocytosis. Okay. Then the next is endoplasmic reticulum, the tubular structure that's extended from the nucleus into the cell membrane. It's called endoplasmic reticulum, and that endoplasmic reticulum can be rough if that endoplasmic reticulum is combined with ribosome, rough endoplasmic reticulum, or that's called granular endoplasmic reticulum, because you can have the granular structure, and these are the ribosomes combined with endoplasmic reticulum. Okay, so rough endoplasmic reticulum involved in the synthesis of proteins because they contain ribosomes. Otherwise, you can have smooth endoplasmic reticulum. Okay, endoplasmic reticulum with no ribosomes is called the smooth endoplasmic reticulum. And the smooth endoplasmic reticulum is very important for lipid formation or lipid synthesis. Okay, lipid formation or the lipid synthesis. Okay, like for instance, look, the smooth endoplasmic reticulum is very important for the following. One, store calcium, like the sarcoplasmic reticulum. SR is called cytoplasmic reticulum. This is organelle found actually in the muscle cell, and you'll discuss this one in your musculoskeletal system. Okay, in the fourth module actually. So, anyways, the smooth endoplasmic reticulum is very important for storing calcium. Okay, and it is very important for the synthesis of steroids, because steroids are made from lipids. Like for instance, you can see sex steroids like progesterone, estrogen in females, or testosterone in male. They made from lipids, and collectively they are called sex steroids. Okay. Otherwise, you can have aldosterone, okay, and other hormones made from the lipids. So a smooth endoplasmic reticulum is very important for synthesis of such hormones. Synthesis of phosphatase. Okay, phosphatase are enzymes. Those facilitate glycogenolysis. I think if you remember yesterday, we have studied that when glucose in your blood is excess, when it is beyond to your body consumption, it should be converted into glycogen and stored in the liver, and that's called glycogenesis. The reverse is called glycogenolysis. That is facilitated by glucagon hormone. That means when glucose in the blood is get less, like for instance, during fasting or when you skip meal, glucose in the blood is get less. Okay, so there should be breaking down of glycogen, and that's called glycogenolysis. Okay, and you can have glucokinase enzyme still made by smooth endoplasmic reticulum, and this is very important for glucose to be converted into glucose 6-phosphate. Okay, and that takes place in the beta cell. I mean that if you remember again, yesterday we have said that there are beta cells in the pancreatic gland. Those produce or secrete insulin. We have said that one. So how insulin is secreted from the beta cell? Because the primary factor is glucose, and that into the beta cell of the pancreatic gland, and that glucose is going to phosphorylate. Okay, phosphate is going to add on glucose, then you'll have glucose 6-phosphate. On the 6th carbon, phosphate is going to add by enzyme, and that enzyme is called glucokinase, and finally insulin is going to released. Okay, so these are some of the functions of the smooth endoplasmic reticulum. You can add more. Okay, you can add more. I mean that more function of smooth endoplasmic reticulum. So we are looking at function of each organism. So nucleus controls the cell activity. Okay. And ribosomes are important for the synthesis of protein. And not only ribosomes, even rough endoplasmic reticulum is very important for the formation of proteins, but the smooth endoplasmic reticulum is very important for the synthesis of or formation of lipids. Okay. The next is mitochondria. Okay, so mitochondria is called the powerhouse of the cell because 95% of ATP or adenosine triphosphate is synthesized in the mitochondria. Look, for instance, the figure. For instance, you can have glucose. The major source of energy is glucose in our body, and that glucose enter into your cell. Okay, and there is glycolysis in the cytoplasm. Glycolysis is a process through which ATP is produced with no oxygen, and that's called anaerobic respiration. So 2-ATP is synthesized in the cytoplasm through glycolysis. Okay, and pyruvate is formed. 2 pyruvate molecules from a single glucose molecule are formed, and that pyruvate enter into the mitochondria of your cell. Okay, and in the mitochondria there are cascades. Okay, and finally are made from single glucose molecule. Okay, so totally you can, I'm just not actually not only 32 plus 2 34 plus 2 36, so 36 ATP made from a single glucose molecules, and that is through glycolysis and oxidative phosphorylation. So oxidative phosphorylation is formation of ATP using oxygen, and that's called aerobic respiration. And that aerobic respiration is because of your mitochondria. Otherwise, I'm not going to discuss about the structure of the mitochondria, like the matrix, the inner membrane, and the outer membrane, which is rough, and the inner is smooth. The inner is rough actually, and the outer is smooth, and so on. So such should be discussed in your anatomy. But at least you should know the function of the mitochondria, because we have said that physiology is a science that studies about the function of the body. So you should know the function of the heart, the function of the mitochondria, the function of rough and smooth endoplasmic reticulum, and so on. Otherwise, the arrangement, the organization, the location, and so on is better to be discussed in anatomy, another science. Okay. The next organelle is or are lysosomes. The lysosomes. Okay. The lysosomes. So lysosomes are circular structure, okay, ring-like structure found in your cytoplasm, and these lysosomes mainly important to digest the foreign bodies that the bacteria enter into the cell, or if there are worn out or completely damaged organelles inside the cell, then that should be digested, break down, and get out from your cell because of the lysosomes. So lysosomes for intracellular digestive system. The lysosomes form intracellular digestive system. For instance, the lysosomes are actually made from the Golgi apparatus. Okay, so the parent organelle for lysosome is the Golgi apparatus. Then you can, as you are looking at from figure 6, there is a bulged out structure from the Golgi apparatus, and then break or bulge off that the bulge structure. Then you can have circular structure like the primary lysosomes. The lysosomes with no foreign bodies. Okay, it's called the primary lysosomes. Okay. Otherwise, like for instance, there is damaged organelle, like one of the organelles in our cell may be damaged or maybe good. So if that's not repaired, it should be removed, and that is because of the lysosomes. So that damaged organelle is going to be taken by or engulfed by the lysosomes and processed, breakdown, then excrete out from the cell, and that's called exocytosis. That's called a kind of bulk transport. Not only the worn out or the damaged organelle that can be taken by the lysosomes, even when the bacteria or virus is going to be engulfed by your immune cell, then after that bacteria or the foreign body is going to engulfed by your immune cell, it should be taken by the lysosomes, and the lysosomes process, okay, breakdown, okay, and then excrete out from a cell, and that's called again exocytosis, okay. But sometimes what is the problem is, if you're looking at the lysosomes, there are membranes, because we are talking about eukaryotic cells, and the inner membrane has protective, because the lysosome contains hydraulic enzyme or there is acidic chemical is found inside the cell, and the inside or the inner membrane of the lysosomes is protected. But sometimes if the lysosomes is damaged, that acid is released into the cytoplasm and kill the cell, and that's called suicide, self-killing process, and that's called autolysis. Sometimes the lysosomes release their acid with no purpose, and that acid is going to damage our cell, and that's called autolysis. There are different diseases result from autolysis. Okay. Then the next, peroxisomes. Okay. Peroxisomes are again organelles found in the cytoplasm, and they are mainly important to produce the catalase. So catalase is an enzyme, okay, that converts hydrogen peroxide, which is very toxic for your body. For instance, if you are going to take more alcohol, then that alcohol make the liver to produce hydrogen peroxide, and that hydrogen peroxide kill or damage the liver tissue itself. So it should be converted into harmless that component, like into water and oxygen, and that is because of the catalase. So catalase is very important enzyme produced from peroxisome. Okay, so these are some of the organelles found in our body with their own function. So that organelles are called the machinery of the given cell. Okay, and we have said that in one tissue there are a number of cells, and those cells found in one tissue should be communicate to each other, okay, through junctions. So junctions can be proteins, or junctions can be ways through which two or more cells found in one cells are communicate to each other. So the first junction is called tight junction. The tight junction. Look, the tight junction. In figure 8 in the right side. Okay. So when you see the tight junction, that means when two or more cells are connected to each other with no protein, rather through their membrane, that's called the tight junction. So no transportation process between the two adjacent cells. So like for instance, if you see your skin, the cells are connected to each other with the tight junction. Okay. The next is called desmosomes junction. Desmosomes junction made by intermediate size filament proteins. So you can have such proteins in two cells. Those need to be communicate to each other. So if two adjacent cells are communicate with intermediate size filament proteins, then that junction is called desmosomes junction. The next is the gap junction. That means if two or more cells are communicate to each other with connexin proteins, you are looking at actually 4 connexin proteins, and then they form gap junction. And if two or more cells are connected to each other with the gap junction, then the two cells or more cells are carrying the same cytoplasm and can be taken as one cell. Okay, and electrical signal can be conducted from one cell to another through this gap junction. You will see this one in excitable tissue or in excitable cells. You will discuss this one in module 1, part 2. After this, after this module, you'll discuss this gap junction in detail, which is very important in your excitable cells, like the neurons of the nerve cells, the neurons of the nervous tissue or the muscles. Okay. The last are adherence junction. If two adjacent cells are connected with actin filaments or actin proteins. Okay. So anyways, you should know that two or more cells found in one tissue should be communicate to each other with cell junctions. So this junction can be the tight junctions, the desmosome junctions, the gap junctions, and adherence junctions. At least you should know this one. Okay. Then there is process. Okay, if you are talking about a given cell, okay, then that given cell needs like oxygen or the nutrient like glucose and so on. Okay, so there should be transportation process across the cell membrane. So transport across the cell membrane is process which substance enter into the cell. That's called influx. When substance enter into the cell is called influx, different from phagocytosis. Phagocytosis is when substance again enter into the cell. So phagocytosis is when substance enter into the cell in a bulk mass. That's called phagocytosis. But in the case of influx, when substance enter into the cell separately, like when sodium enter into the cell, it is called influx, not phagocytosis. But when bacteria is going to be engulfed by a given cell, that's called phagocytosis. Okay. When ions leave the cell, that's called efflux. Okay. So membrane transport is a process through substance enter the cell, that's called influx, or leave the cell, that's called efflux. Okay. So the passage or that process can be through the lipid bilayer with no proteins or with proteins. Okay, so it can be through the lipid bilayer with no proteins, or that can be with proteins. Okay, sorry, let me interrupt. Sorry. Just a moment. Sorry. Okay. Sorry, guys, for the interruption. So the passage or the process of influx or efflux can be through the lipid bilayer, and just with no proteins, or it can be through the proteins. Okay, and it knows that the permeability of one cell is different from the other. It's obvious. For instance, the permeability of the cell membrane of one is different from the other because of variation in composition and the variation in the arrangement of the composition. Okay. If you see this one, the transportation process across the cell membrane can be with no protein. Like for instance, if you see oxygen transportation through the cell membrane is through diffusion. Okay. Or if you check like the gas, the carbon dioxide, or the liquid soluble substances. Okay, so gas, liquid soluble substance, alcohol, and so on enter or leave the cell with no protein. Otherwise, if nutrients like for instance like glucose, amino acid, or iron, water, and so on need to enter to the cell or leave the cell, proteins will be there. So the transportation process can be with no protein, that is through the lipid bilayer, or can be through proteins. So it is good to know what are substances enter into the cell or leave the cell with no protein, and what are list of substances need to enter into the cell or leave the cell through proteins. It is good to know that one. Okay. In such a way that the membrane transport can be passive or can be active. So you can have passive transportation process if the influx or efflux of substance across the cell membrane doesn't need any energy. That's called passive transportation process. Okay. If that process doesn't need energy, that's passive. And why that transportation process doesn't need energy? Because substance move from high concentration to low concentration. So the movement is called down concentration gradient movement from high to low. For instance, if you need if you need to leave your pen into the ground, no need to apply energy. Okay, the same story here, and that is because of the concentration and electrical gradient. For instance, we have said that sodium, calcium chloride concentration outside the cell is much higher than the inside. So sodium influx is passive or active? Tell me. Sodium influx is passive. Calcium influx is passive. Chloride influx is passive. Influx is passive because the concentration of this substance outside the cell is much higher than the inside. Okay, so when substance move from high to low, energy is not required. Okay, or that's called downhill movement. Okay, downhill movement, or top to bottom movement, or high to low movement. You can say. Okay, so it is a concentration gradient that drives substance to enter into the cell or leave the cell from high to low. So no pressure gradient is required here. So no energy is required here. So if the transportation process doesn't need energy, that's called passive. And you can classify the passive transportation process across the cell membrane into simple diffusion, okay, facilitated diffusion, and osmosis. So the first is called simple diffusion. So in the case of simple diffusion, substance move from high to low through the lipid bilayer or through non-gated ion channel proteins. Non-gated. Non-gated proteins are proteins lack gate extended from the channel. Channels are proteins. So when substance enter or leaves the cell through a lipid bilayer or through the non-gated ion channel, that type of passive transportation process is called simple diffusion. Like, for instance, lipid-soluble substance like vitamin A, vitamin D, and others enter into the cell or leave the cell with no energy, okay? And that is through the lipid bilayer, so it is through simple diffusion, or the gas, alcohol in its own, okay? But if that passive transportation process is facilitated by a special protein that's called permease, it's called facilitated diffusion, okay? So in the case of facilitated diffusion, solutes or substance that is required to enter into the cell or leave the cell should bind on the binding site found on the carrier. No binding in the case of simple diffusion. Okay, no need binding. So that means substance that needs to enter into the cell or leave the cell is not required to bind on certain protein. No, but in the case of facilitated diffusion, there should be binding. So there should be binding site. So once the substance that needs to enter into the cell or leave the cell is going to bind on the binding site on carrier protein, then there is conformational change, like rotation, opening of the protein. The protein may rotate, okay, or may open, so that substance enters or leaves the cell. Substance released into the low concentration, because we are talking about the passive transportation process. So substance released from high to low, or high concentration to low concentration. Substance attached from high to the concentration, from high concentration region into the low concentration region. Sorry. And in the case of passive transportation process, there is what you call diffusion rate, because in the case of passive transportation process, there is what called diffusion. Diffusion is random movement of substance from high concentration into low concentration. That is called diffusion. And different substance, when cross the cell membrane, they take different time. Okay, that's called diffusion rate. Time taken by substance to cross the cell membrane is called diffusion rate. So the diffusion rate of oxygen is different from the diffusion rate of carbon dioxide. The diffusion rate of vitamin A is different from vitamin B and so on. So there are so many factors affecting the diffusion rate of a substance across the cell membrane. And these are some of them. So I listed down 7 factors affecting the diffusion rate of substance across the cell membrane. The first is the thickness of the cell membrane. So the more the thickness of the cell membrane, the more the time taken by substance to cross that membrane. So the diffusion rate is less. You should know how the thickness of the membrane affects the diffusion rate of a given substance. Okay. The next is the molecular size. Okay. The smaller the size, okay, the smaller the size, the faster the movement. The faster the movement, there is a time taken by the substance to cross the cell membrane, so that its diffusion rate is high. Okay. The third is the temperature. The third is the temperature. Okay, the temperature. So the higher the temperature, the higher the diffusion rate, and the higher the diffusion rate is the time taken by that substance to cross the cell membrane. That there is a gradient difference. Okay, the concentration gradient difference. The more the difference, the more the diffusion rate. For instance, if you see the number 1 and the number 2, the gradient or the difference between the cell membrane is high in number 1. So the diffusion rate of the same substance in number 1 is much higher than in number 2. Okay. The other is the lipid solubility. The more the lipid solubility of the given substance, the more the diffusion rate. Okay. And the other is the surface area available for diffusion. The more the area, okay, the more the area for diffusion, the more the rate. The more the rate, I mean that the more the diffusion rate is, the less the time taken by the substance to cross the cell membrane. And the last is the permeability of the cell membrane. So if the cell membrane, permeability is ability of the cell membrane allowing to pass a substance is called permeability. So the higher the permeability, the higher the diffusion rate. The higher the diffusion rate is, the less the time taken by that substance to cross the cell membrane. The last type of passive transportation process is osmosis. So osmosis is the net movement of water across the cell membrane. As you know from your biology, I think, osmosis, the net movement of water from high water concentration into low water concentration, or a solution with high water concentration is called hypotonic solution. So osmosis is the movement of substance from hypotonic to hypertonic solution. Okay, or from low solid to high solid concentration. Okay, and this osmosis tries to equalize amount of water found inside and outside the cell. Okay, so osmosis is until equilibrium is maintained between the two compartments. Okay, so if there is water and solid concentration, osmosis is there. Otherwise, if there is no water and solid concentration difference between the cell membrane, no osmosis at all. Okay, because osmosis is net movement of water. Okay. Otherwise, there is tonicity. I think you'll see this one again in your hematology, but it is good to have some concept about the tonicity of your blood. So your blood can be isotonic. That is, for instance, if the concentration of intracellular fluid and extracellular fluid is the same, then that solution is called isotonic. Okay. Otherwise, if the concentration of the blood, okay, is less than the concentration of fluid inside the cell, then your blood or that solution is called hypotonic. Otherwise, it's called hypertonic. Okay, against to intracellular fluid, or that's called cytosol. So this tonicity always affect osmotic pressure of that a given solution. Okay, for instance, if you are going to put a cell in isotonic solution, what would happen to the cell volume? What would happen to the cell volume? Nothing changed. No change at all. Okay? But if you put a cell into hypertonic solution, the cell volume is reduced. It's called shrinking. It's called flaccid. Why? Because if you are putting a cell in hypertonic solution, more water is found inside the cell than outside, so that water leaves the cell. So the cell volume is going to affect it. Okay. Otherwise, if you put cell into hypertonic solution, water enter into the cell. Okay, water enter into the cell. So when water enters the cell, then the cell volume is going to increase, blood and so on. So anyways, the tonicity of your blood or the tonicity of solution you prepared affect the cell volume, like the red blood cell. Okay. Otherwise, there is what called osmotic pressure. Actually, we discussed the standing force yesterday when we discussed about the filtration fluid at the arterial end of the capillary. Okay, we mentioned that one. So osmotic pressure is a pressure created by solutes like electrolytes or protein that oppose movement of water into another compartment. It's called osmotic pressure. Okay, osmotic pressure, and that osmotic pressure is affected by the tonicity of solution or affected by the osmotically active particles in a given solution. Like 1 mole of glucose has 1 osmol per liter, and 1 mole of sodium chloride has 2 osmol. Why? Because the sodium chloride can be dissociated into sodium and chloride ion. So 2 osmol. 1 mole of hydrochloric acid, 4 osmol. And 1 mole of sodium phosphate, 3 osmol, because it can be dissociated into 2 sodium ion and one sulfate ion, 3 osmol. So from this molecule, which of them that has high osmotic pressure? Sodium sulfate, because osmotic pressure is affected by osmotically active particles. So it is sodium sulfate that contain more osmotically active particles, the 3 osmotically active particles than the other. So this osmotic pressure is affected by osmotically active particles in a given solution. So the more osmotic pressure is, the less the movement of water into another compartment, because osmotic pressure is because of the solid like sodium or protein and so on, allowing water to leak out into another compartment. And the size of the molecule, again, the size of the molecule doesn't affect osmotic pressure. Like for instance, if you see the size of glucose is greater than the size of sodium, actually, if you calculate the molecular weight of glucose, you can like for instance 6 times 12 plus 12 times 1 plus 6 times 6 times 16 and so on. Anyways, the molecular weight of glucose is greater than the molecular weight of sodium chloride, but it is sodium chloride that contributes high osmotic pressure than glucose. So osmotic pressure not affected by the weight. Okay, and the chemical nature. Okay, and it can be expressed in terms of osmolarity of your blood. Okay, so the last, okay, we left with some slide or one or 2 slides, sorry. So please wait for some time. The last is active transport. So in the case of active transport, energy is required. Why? Because substances are expected to move from low to high. So when substance enter into the cell or leave the cell, from low to high energy is required. So in active transport, proteins should be involved, and those proteins are mainly the pumps, like the sodium potassium pump. We mentioned this one when we discussed about the intrinsic homeostasis control system. We mentioned this one, and the calcium pump. Okay, so pump mediated, the pump mediated like the bulk transport, like exocytosis or endocytosis. So exocytosis, endocytosis are active because they need energy, or when substance move from low to high, that's called against concentration gradient, or that's called uphill movement that need energy. So such kind of transportation process across the cell membrane is called active because energy is required. And the source of energy is directly ATP or ions. So active transport can be divided into two, okay? Primary active transport or secondary active transport. So if the source of energy is directly ATP, it's called primary active transport, or that's called the direct active transport. Otherwise, if the source of energy is not directly ATP, but if that is ions, it's called indirect or secondary active transport. Okay. Otherwise, in the active transportation process, we need proteins. Okay. Uniporter, symporter, or antiporter. I told you this one in the earlier slide, so that I'm not going to repeat it. Okay. So active transport can be divided into two based on the source of energy. So if the source of energy is ion, it's called secondary, okay, active transport, or indirect active transport, or ion driven active transport. Otherwise, if the source of energy is directly ATP, it's called ATP driven, okay, active transport, or you can say primary or direct active transport. So in our body, both type of active transport are found. Like for instance, in the case of direct active transport, the source of energy is ATP, so that ATP, that is adenosine triphosphate. There are three phosphate combined with adenosine. So when one of the bond is break, energy is produced, okay, and that energy move substance from low to high, or that's called against concentration gradient. Okay, and that breaking down of phosphate is by enzyme, and that enzyme is called HPS enzyme. So HPS enzyme is very important for the hydrolysis of ATP, and energy is released, and that energy moves substance from low to high. Okay, look this one. Look the sodium potassium pump. We mentioned this one yesterday. When 3 sodium leaves the cell, 2 potassium enters the cell. So when sodium leaves the cell, it is called efflux. So sodium efflux is active or passive? Tell me, please. Sodium efflux is active because sodium inside the cell is less than the outside, and sodium is moving from low to high, and that need energy. And the source of energy here is direct ATP, so it's called primary or direct active transport. One example in your body. You'll see this one in your physiology courses, in the coming physiology courses. But so it is good to have some concept about the sodium potassium pump. So when 3 sodium leave the cell, 2 potassium enter into the cell. So this sodium potassium pump is very important. The cell volume, I told you this one yesterday. How sodium potassium pumps regulate the cell volume. I told you, if you remember. Create membrane electrical gradient. Yes, because sodium side is the net charge inside the cell is negative and the outside is positive, because 3 sodium leaves the cell and 2 potassium enter into the cell. So the cell is losing 1 cation through 1 sodium potassium pump. So if there are a number of sodium potassium pump, the cell is losing a number of cations so that the inside become negative. So that's called electrogenic effect of sodium potassium pump. So electrogenic effect of sodium potassium pump is ABCD. You should know why we call sodium that electrogenic effect. You should know that one. Maintain ionic diffusion gradient. Okay, that means when 3 sodium leaves the cell, sodium is highly accumulated outside the cell, and sodium may enter into the cell through diffusion. That is sodium influx, which is passive, and that is because of the sodium potassium pump that try to accumulate more sodium outside. Okay, and contribute. Like for instance, when we go to the cell from low to high, look figure 12. So like you can have 3 glucose molecules inside the cell and 1 glucose outside the cell. So sodium is entering into the cell from low to high, and that needs what? Energy. And the source of energy is not directly ATP, rather sodium. So sodium enters into the cell from high to low. So energy is not required, rather energy is produced. If you remember from your analytic chemistry high school, when electrons that get down from higher excitation state into the lower excitation state, energy is produced, exothermic reaction. And that energy, the same concept is here. So when sodium enters the cell from high to low, energy not consumed, rather energy is produced, and that energy drive glucose from low to high. And that's called secondary active transport. One example. You can mention some other. Okay. So this is all about the introduction in the cell physiology shortly within today's lecture, or 3 hours, 6 hours, sorry. So I have put my email in the chat box, so you can communicate with me. Okay. So when you get some difficulties, okay, you can email, and I will respond for you. So thank you very much. But there are that student who left the lecture. That's not good. Okay, one of the ethics expected from the medical student is to attend the lecture. Okay, can have the screenshot to attend the lecture. You are adult, you know which is important, so that it is good to attend the lecture than reading many or different books. It takes much time, or it may be difficult to understand. So attending the class is very important. You may gain something, okay, from the lecture or from the experience of the instructors, okay.